Okay, so the story that I want to talk about today is the interaction between proteins and nucleotides. And I want to talk about it ultimately because of two different reasons. One is protein binding or protein interactions. When you ask what proteins bind to, do you have any idea how many different kinds of chemical molecules proteins bind to in the space of all organisms? If you counted all the possible chemicals that proteins bind to under natural conditions, any idea what the number would be? Compounds, so we're not talking about atoms, we're talking about molecules or compounds. How many things or any life form out there, how many things do they bind to, do you believe? Any idea?
What do they not bind
to? That's an unfair question. So I meant, let's just begin, stones. Okay?
That's not part of an organism.
Well, so there's tons of chemical materials out there that are part of an organism, so they don't bind to it. So if you now made a registry, if you just counted all the things that they do bind to in natural situations, how many things would that be? How many molecules? So for me, very surprising, the answer is just in the ballpark of roughly over 1,000 or something like that, between 1,000 and 2,000. So the final count there is still out, but it is a remarkably small number in some sense if you look at the space of all possible interactions. Again, it's difficult to, now you're sort of getting in a circle, right? You say
there are organisms who bind to stones, and I think their proteins are part of this binding mechanism.
Good point. All right. Arguably, so I don't believe that you will argue with me that the space of all chemicals out there is way bigger than thousands, right? Way bigger. Different chemical compounds. I do not actually know what this number is, but this is certainly in the 10 by the 6. It's certainly in the million, and most likely many millions. And when we sort of sample all the known organisms that we know, maybe some of them, under some conditions, actually bind to many of those. We don't know. But so when we simply do a count of all the kinds of things living organisms use for their life, for the regular life, then that number is surprisingly small. Again, you may be aware that there are bacteria that eat oil spills. But oil is not typically a natural product of natural evolving organisms or something like that. Again, this is debatable. And it's also debatable when you call them natural, when you call them designed, and so the story gets complicated. But whatever. So let's just turn this question around. What are the most important chemicals that things bind to? And this is a few thousand. And in these few thousands, the largest molecules proteins bind to are proteins and actually nucleic acids. DNA and RNA. So those are the largest things they bind to. Some of you may remember this plot here where I pick a residue that I know binds to something, in this particular case another protein, and I ask, in the sequence environment of that, if the central residue is a protein binding residue, and I go left 4 and right 4, and these 9 residues, how many binding residues, protein binding residues do I find? Blue is, I define binding residues within less than 6 angstrom, yellow within less than 4.5 angstrom, or 4 angstrom. And just at the blue you see that in this window, most of the time, there are sort of 6 to 7 binding sites. Any binding residue is surrounded by many other binding residues in the sequence environment, right? So we are not talking about this sort of enzyme story where we have 3 binding sites that are far away in terms of sequence space. We're talking about a situation where there is like this somehow here. You have a binding of 2 blue molecules here, and there are many sequence consecutive residues here that bind. That's the situation for protein-protein, and that is also the situation for protein-DNA. Give me some examples. So the major minor groove. So here's an example of the lumbar repressor. that binds into two parts of the DNA. So here we have the first example of something where you have a sequence, relatively sequence consecutive element, where you have a lot, this helix wraps into this groove, and in fact many of these residues in this helix bind to DNA. And these are adjacent residues. Okay, it's I and I plus 4, but you see that in a window of 10 residues, several of these 10 here are binding. Okay, here we have the example of the nucleosome or the histones, where proteins wrap, or DNA wraps around these, and that in fact is again the similar situation. So you have a lot of DNA packed into a relatively small protein space. And here's another example where you sort of have a pincher, but even in the case of the pincher, the leucine zipper kind of motif, even in the case of the pincher, you still have, in this particular case again it's a helix, you still have a relatively local binding site, so many residues binding in the local, in the binding interface. And let's just leave it there. So that brings us to the question, how many proteins in human? We have 20,000. How many bind DNA? And this is a question that I cannot answer. I do not have an answer for that. So we have some answer. I will flash a number that I'm no longer really trusting in. What people do look at are transcription factors. Do you have an idea how many transcription factors we have? So transcription factors bind DNA. So clearly the number of, if I give you the number of transcription factors, then the number of proteins that bind DNA is higher than that, right? That's it's a lower limit. Any idea, ballpark? Is it hundreds? It's not 10,000, so we are all clear about that. And it's not 10. The number is somewhere in between these two extremes. No? Nobody would be shocked if I said 5,000. Would you guys be shocked? Too much. If I said 50, somebody shouting, this is too little. Yeah, it's too little. Okay, we're getting closer. So what if I threw 100 at you? Too little? Or it could be? Are we now talking it could be? And 1,000 could be too? So, I want from you that you're within an order of magnitude. So if you're sort of not deciding between 100 and 1,000, that's already good, right? You say 500 and in your estimate you're within an order of magnitude. So if the true number is, again, is quite unclear. So the most recent paper that I just found, typically I only quoted this paper here, but I just yesterday found another paper that is more recent. It comes from looking at more detail of higher resolution data. They are not incompatible, so these are 2 points that are compatible. They said more than 1,391, and the others say about 1,600, and these two numbers are compatible. So that's the ballpark. So round about 8% or so of the human genome would be transcription factor, which I believe, for me, is a surprisingly large number. But again, so this seems to be what people, and again, there are arrays out there where people actually put, as an experimental array, something in the ballpark of 1,000 something already onto one array. So people have their hands on more than 1,000. So this is also clearly not an overestimated number. Right? This is sort of the ballpark where we are sitting. So from 3D data, how do we get to protein prediction methods? So one way is when we look at the three-dimensional structure, the simplest way we can do that is we can simply identify the nucleotide, identify the amino acid, and say if they are within a certain distance, just like we did for protein-protein. We call it a binding residue, right? And for Francisco Melo's group, I can't remember Tomas's last name. You can read it. Anybody speak Spanish? No. Not good. Anyway, so Tomas and Francisco Melo developed this database here that essentially defines the interfaces between DNA and RNA for proteins in the protein database, in the PDB. And the way they do it is first they look at this proximity issue, but then they also look at energy. The energy of binding. Why is that relevant? Well, one thing is DNA is very negatively charged. So you could imagine that you have, in fact, a very positive positive stretch here that falls, say you picked the 6 angstrom, or you picked the number 4 angstrom. So you said that if the closest atom between an amino acid and a nucleotide would be within 4 angstrom, I call it a protein DNA interaction site, right? So that would say, put the limit here. So by this you would say that these residues interact. Now, maybe you see a very positively charged stretch near it that is not within the 4 angstrom. But since that is contributing clearly, say you had an R and a K here, then that would create a local pocket of positive charge that would create an electrostatic potential that, although this may be 10 angstrom, would create a local field that would attract the DNA. So that would most likely be relevant for the binding. If I want to predict the binding site, I actually would want to know that. Because, for instance, if I evolutionarily change that, I would see an effect. The binding would no longer happen. Even if I evolutionarily change it, if I change it in an experiment, you may no longer see the binding affinity as strong, because you create a potential. So if you want to create a binding prediction method, then this should be taken into account, right? And that's in fact what they do. Now, let's just keep it there. There's an interface and there's a database. Since we need that for DNA, why don't we do that for proteins? Why don't we need it for proteins? Do we need it for proteins? So far we have never spoken about that. Is that just something that I hid from you? Sure it is. It's something that I did not tell you about. So I hid it from you. But did I hid it because it's important, or although it is important, or did I hid it because it's irrelevant? The problem is a different one. The problem is we essentially do not know the answer to my question. So I take it that you all not contributing an answer to this point means you don't know the answer, and that's a good answer. That's the right answer. So we all don't know it. So we all share this. And ultimately this is because doing the molecular dynamics for protein-protein interaction is very complicated. Because of the entire protein, we essentially don't understand the complexity of a protein. Understanding the complexity of a piece of DNA is much simpler. DNA is simple as an object that you simulate in a molecular dynamics simulation. You can't fold DNA. You cannot fold proteins. Okay, that ultimately is the background here. So we can afford to do that for protein DNA. We cannot afford to do that for protein-protein because our understanding is not good enough. Or maybe we can afford to do it, but it's sort of more tricky. Let's put it like that. Okay. So then the first question is, what are the methods that exist? Or no, the first question is how do we actually do it? In fact, this brings us to methods that exist. There's a historic motive kind of approach. This one goes back to Janet Thornton. That is helix-turn-helix motifs. So several of those motifs that I showed you are the traditional binding motifs. Then there are charges and dipole moment kind of methods that use 3D structures to predict it. A couple of those are shown here. That is the grasp method. Then there is an approach from Nirbental's group, or was an approach from Nirbental's group that used 3D structure and conservation signals, so essentially looked at the alignment features, so the pieces M, so to speak, but visually on a 3D structure, and combined that in a clever algorithm. Now, there are then other things that we need to do in order to do that here, because ultimately, in order to find such a local environment where positive charges can influence the DNA, I need to find patches on the surface. Patches on the surface, ultimately, in this case it's easy. So I constructed a case where R and K are close to each other. They will create a positively charged environment, right? If you had a situation such as this one, where a positive charge would sit here and another one here, whether they together contribute to a strong field or not, depends on whether they find each other in 3D, so they are together on the surface. And that needs, in order to do that, we need to find patches. So parts of the surface that act together and have a similar field, and this is what is ultimately the major theme in this one here, finding patches from 3D structures. This is done through machine learning. It works fairly accurately, and you can use that in this particular case. However, they have been using it only for really DNA. So this, you could imagine now these patches would be relevant for all kinds of function prediction. It worked actually to the extent it worked only really for DNA binding. And it allows you to predict, for instance, active sites. Now, in a more from sequence, typically one way in which we can do it is nuclear localization signals. I briefly mentioned that in context of the localization prediction, that we have this mechanism of nuclear localization signal where the protein binds to it and brings the protein into the nucleus. Most proteins in the nucleus bind DNA. So the same motif that binds to the protein binds to DNA. Here's an example. So the one that is labeled here is exactly the nuclear localization signal. So exactly the motif that binds to protein. And the same you see binds to the DNA. Right? This is exactly what fits into the groove here. That then you can use and simply take all the nuclear localization signals that are known, and you look at the subset for which DNA binding is known, and you call that the NLS DNA binding motif prediction method. Okay, and that is one simple method. And that simple method for human found about 400 proteins that are sort of NLS-based predicted to bind DNA. Another way of doing it is you do a machine learning device, and this machine learning device, again, you start with a data set such as the one that is developed by Francisco and Tomasz. And when we did that initially, we is Janai Ofran here, and Venkata Mysore, with help of Trevor Siggers. Then we got a view on performance that was remarkably good. So here's the per residue performance. So this is accuracy, meaning precision, versus coverage, recall. And the blue ones here are the per protein level. So per residue simply means which of my residues binds DNA. Per protein, can I distinguish between a protein that binds DNA and a protein that doesn't bind DNA? Okay. Now, by the way, the background data set is we have 250 proteins that bind DNA and 291 protein DNA complexes, but for some of those we actually don't know the binding site. And those are essentially what we use as negatives are those. Okay. And it appears here as if we are very good because for 50, wait a minute, the axes are labeled incorrectly. This axis here, oh, I have to change that, is recall, and this axis is, no, it's right. No, the axes are wrong. So I have 100% precision for 50% of my cases. So the axes are wrong. The axes are right, you have to just flip them. In some sense they're right, in some sense they're wrong. They're totally wrong, I'm sorry. So it seems as if for 50%, the 50% best predicted, we make no mistake. And that, in fact, is what this here suggested, but this is in fact absolutely wrong. And it turned out there are several mistakes we did. Number one is we did not sample enough proteins that really don't bind DNA. Okay? Number two, essentially for us in this universe, everything that didn't bind DNA in the DNA binding protein was a non-binder. And that is not enough. The second thing that we did is we looked at data where we had the 6 angstrom plus the MD data. Okay, so let me see whether we can figure this out. Let's call it approach 1. In approach 1, I take bind and non-bind, and I distinguish by RMSD plus energy. In my next approach, in approach 2, I look at bind or not bind, so I don't have to repeat that, and I only look at RMSD. What do you believe would be the difference between the two? And again, let me repeat. So for the story of protein interaction sites, we only ever looked at this part. Now, I have a machine learning device that simply looks at energy plus RMSD and trains on a per residue level between residues that bind DNA and residues that do not bind DNA. Residues that bind DNA are those that fit the RMSD and the energy criteria. Residues that are labeled to not bind DNA are those that fit neither. So they are far away and have no contribution to energy. Everything in between is thrown away. Okay.
In the training data set.
In the training and testing set. Oh, by sort of the movement of my head was misleading. The answer should have been no. So in the entire data set. And that entire data set is then cross-validated. Can anybody see? What do you expect if... so here I use that, and here I use only RMSD. What do you expect the difference to be? Okay, if I asked you, which one is easier to do for a machine learning device? The first approach, where I essentially look at RMSD plus energy, or the second approach, where I look only at RMSD?
The second approach.
Why?
Because you have less data to input to the...
You have less data? You actually have more data.
You have less dimensions.
No, less dimensions.
I'm sorry, you have
less dimensions because...
Oh, no. It's only 2 dimensions. I'm not entirely sure what you mean by dimensions. I only learn whether it binds DNA or not. My mapping problem is not different. My input is the same, my output is the same. All I change is my data set. So how do I have less dimensions?
Well, you don't have to calculate energy.
No, it's much easier to do for me. If I asked you which one is easier for you to put into your thesis, absolutely number 2. Sure. 120% true. But I'm asking, what do you expect in terms of the machine learning? What do you expect to be easier for the machine learning? And it turns out that the first one is much easier. The reason simply is that you clean up the data. By throwing everything in between away. What you throw away are all the ambiguous cases. What you keep are the very clear cases. The ones that absolutely clearly bind, the ones that absolutely clearly do not bind. Uncertainties fall out. That's easier for the machine learning device than keeping the ones in between. Because keeping the one in between means that sometimes you make sort of an error. You experimentally see something as DNA binding that actually is not, and that's difficult to learn. And vice versa, right? But you clearly keep cases that require more information, maybe environment dependent. Maybe the residues that would bind if the environment would be different, right? So this is a cleaner data set. Now, sometimes in machine learning, giving a cleaner data set is good. Because by a cleaner data set, you have better training. So now back to your question, do I use it only for training? Sometimes it could be the right thing to do to train on a cleaner data set. And sometimes not. And the difference has to do with how complex the problem is, how many you have. How many do you actually throw away? In this particular case, we threw away many. That could be a disaster. In this particular case, the major thing was not, I believe, that we threw some away. The major thing, not in terms of training, the major thing was that in terms of testing, we cleaned up the situation too much. That means we made we made the problem simpler for testing. We only looked at the ones that had very clear labels, and that allowed, in some cases, to reach really high levels. And Jajun is going to show you that our current best method is by far less good. And this is one of those things that happen to people such as you, who come and develop the 15th method that exists or something like that. Very often all that you can do is say that everybody so far overestimated. I do the best, and I actually, on my new data set, I show that my method is better, but it's still worse than anybody published. And that really doesn't make it easy to publish. Okay, now one thing that we have to do once we have a method, and we have done that only for this method that we have for a few years here, is we simply apply it to an entire data set. Unfortunately, I do no longer have the label here what this is for. I believe this is in fact for Swissprot. I believe this is the fraction for which of proteins for which DNA or RNA binding is known. And now I'm asking for how many of these proteins for which somebody says they bind DNA, do I actually know which site is the binding site? And this fraction here, the red one, is the ones where I have it through some motives. So that's some database that says this is because HTH or something like that, right? And the green is the fraction for which we actually do it by prediction. And the same thing now for different organisms. Unfortunately, I no longer have the organisms here, which ones those are. Oh no, wait a minute, this is not for different organisms, the different slices in terms of accuracy. So completely wrong. So if I only take the ones where then we believe we make no mistakes, so the most reliable ones, then this is sort of the fraction of new versus sort of explained by motif. And if we look at the cut of less accuracy, then we essentially get a slot like this. Now we would have to apply that to an entire organism. Again, I haven't done that. And the next generation method, and I just want to flash this, so it started with Michael Mendel, who actually is now in Helmholtz, has done his PhD in Cambridge at the EBI, and is now in Helmholtz. So he was the first who redid this, and the second who then redid it is Peter Hönig-Schmidt, who is about to do his PhD in Weihenstephan. So Martin, you may know him. Have you ever actually seen him?
Yes.
I thought he was sitting in. Anyway, this is not for the camera. So Peter's important contribution here was that he had this insight. Okay, I want to have a DNA prediction method, and actually I want an RNA prediction method too, because RNA ultimately is the other big molecule that proteins bind to. And maybe by sort of combining this, I can benefit from it. Maybe by weaving these two methods into one common framework, I can benefit. And that, in fact, is what Peter showed in his master thesis, that by having a hierarchy, he can improve over a single method that does only one of those two. So in his master thesis, he developed a method that sort of had a first step that essentially predicted, he called that method SUM-NA, some nucleic acid. And the point is that the first step in his prediction was really a machine learning device that said it binds some nucleic acid or not, either RNA or DNA. And that was a very important pre-filter for getting the best DNA and the best RNA prediction method. Okay? So you have one that sort of combines them both and pre-filters the data, and then there's the fine-grained filter, DNA and RNA. And Jajun is going to come in a few minutes and present a different view on that. There are a bunch of methods existing, and again, this is DNA only here. Janju is going to present some of those. Almost all of those have been developed primarily on proteins that bind DNA. So almost all of those fail to distinguish between proteins that bind DNA and not. So all of these focus on if I have a protein that I know binds DNA, where is the DNA? Which residues bind DNA? These kind of methods do that, and there's a variety of methods underneath. I just put the list up. Again, I'm not expecting you. Obviously, I'm not expecting you to remember this page number here or any name. But I do hope that you get away with is the idea that there's a bunch of methods that do it, and they have a problem. Now, all of these methods, they predict that a particular residue binds DNA. How could I come from that prediction to the prediction that a protein binds DNA? In a very, very trivial way. Let's call it hack way.
So you predict if amino acid binds DNA.
Yeah.
Then you just check if a lot of those amino acids are close together in the protein.
So if I simply see, what do you mean by close together?
That's an interesting question. Depends if you have the 3D structure, you would look for...
We don't.
Then you have to look for the sequence and hope the best.
So you somehow, if I had an energy score here, DNA binding, and what you talk about is sort of, this is not binding or something. So then the question is, which peak do I filter out, right? But ultimately, yes, that's exactly one way of doing it. An alternative way of doing it is you would simply ask, if I summed up over all DNA binding in one particular protein, and the sum would be smaller than 5 or 10 or whatever the number is, then the protein is not binding DNA. It's somehow like what you suggest, but I would argue that what you suggest is sort of is cleverer. Because you run a local filter, because you I would clearly argue if you had 20 peaks of one residue, they would add up to 20 residues binding DNA, but this is nonsense, right? Local peaks, there's no protein known that has sort of 20 completely isolated binding sites. So in that sense, this method is clearly better than one that simply counts the number of residues that bind. So these, historically, in fact, what we did is not your intelligent way. Historically, what we did is the dumb way. We simply asked, is the number of residues predicted smaller than 5 or some number like that, or 10, can't remember. If not, then we simply say this protein is not binding DNA. Now, again, RNA interactions. RNA is the other macromolecule. It sort of looks like DNA. What are the major differences between RNA and DNA for you? There's an R where there's a D in the other one. That's a major thing. It's a different nucleotide. Yeah.
There's an hydroxyl group.
Yes. Yes.
One atom I don't know which I have to put into.
Yes, but on that level of structure, yeah. Yeah?
They can build secondary structure.
Yes, why? Why can they build secondary structure? Which ultimately, biochemically, we are getting back to that point. But there's another feature, which is the flexibility, ultimately. They're more flexible, and that allows them to bond to each other. They create structures, and importantly, they function, right? So they act as enzymes. So they have some enzymatic activity. One example here is the ribosome, where the blue ones are the proteins here, and everything else is really RNA. So you have RNA that essentially is absolutely crucial to make this complex here work. Here's the secondary structure story that you talked about. In some cases, actually, the RNA binding site is also the nuclear localization site. So there are examples. What I did not tell you when I showed you this example is this, in fact, could have been RNA, because this particular nuclearization signal does in fact also bind RNA. But when we started this work here in 2000, people had published work that said essentially RNA and DNA binding sites are alike. So yes, there is a little bit of difference, there are a little bit different preferences, there's a little bit of more variation, so they are slightly different lengths, but essentially, in many cases what binds RNA can bind DNA. Today we know, 18 years later, we know this is essentially not true. And we know that these binding sites are rather different. What they are similar in, they're both very extended. So the type of binding is again not the enzyme-like binding, so it's not isolated site in the protein, but it's elongated stretches that bind, just like in proteins. Okay, there's a bunch of prediction methods, and I believe I'm just going to stop here.